Ladies and gentlemen, good afternoon. I will start with the topic of combination of new biotechnologies with hyperbaric medicine. This is just to give a simple scheme of important new technologies and where they are placed in regard to neurological disorders, which is the focus of my presentations, plus hyperbaric oxygen. So this scheme shows how all these technologies are connected to both of these, either directly or indirectly. Now, you can see the cell therapy can be facilitated by hyperbaric oxygen, and cell therapy is being investigated now for neurological disorders even without adding anything. Biomarkers are very important in trying to determine not only the diagnosis but the effect of treatment in neurological disorders. And if you take technologies like nanobiotechnology, it has direct application on neurological disorders. It also provides refinement in biomarkers and molecular imaging. As you know, molecular imaging is very important for evaluating hyperbaric oxygen, particularly in the brain. And we have gene expression, for example. There's the effect of hyperbaric oxygen on gene expression. When you put together all of these technologies, some or other help to personalized medicine or development of personalized medicine, which is the trend these days, that we are stepping into an age of, you may say, personalized medicine. Already it's being practiced in some institutions in USA, not universally accepted as such, but the concepts are well recognized. So the overall impact of hyperbaric medicine on this topic of, if you want, the various ways, such as molecular diagnostics. I mentioned biomarkers, but molecular diagnostics, the best known of which was polymerase chain reaction, but there are many other new diagnostic technologies that are being introduced. Now do we understand the molecular base of disease much better, but we can also make earlier and more precise diagnosis. Now, sequencing of the human genome is already completed around the year 2000. That has a considerable impact on development of molecular medicine. So we are actually in a post-genomic age. And many other therapy, going beyond that, biological therapies. By biological therapies, I mean therapies that use living materials such as cells. We have, if we use gene therapy, we can also use genetically modified cells or viral vectors. So they are in clinical trials and they interact with hyperbaric oxygen therapy. Now, cell therapy, if it is going to use one's own cells has been around for a long time. There are many immunotherapies for cancer based on preparation made from the patient's own tumor cells. So they were sort of personalized therapies. You can call them immunotherapies, and they're also very relevant to HBO, which also has action on the immune system. Plus, we will see other connections between cell therapy and hyperbaric oxygen. So it's not only the clinical applications that are being explored, new biotechnologies provide us methods that have refined our tools for research, basic research. So there are many applications strictly being developed for laboratory research using nanoparticles or many of the cell therapy technologies that will indirectly have an effect on clinical medicine, but not immediately. Nanobiotechnology, as I mentioned before, not only improves diagnosis, it also facilitates the delivery of therapy. Now, drug delivery is not a problem. Hyperbaric oxygen, we are delivering it in a different way. But for other pharmaceuticals, nanobiotechnology has made tremendous contribution to improve the targeted drug delivery. So the reason I talk about this is because I think that we will have personalization of hyperbaric, personalized hyperbaric medicine in the same way as the rest of the medicine, because it is a part of medicine, so far as I'm concerned. Now, I'll just say a few words about nanotechnology. It seems far-fetched. How would nanotechnology be connected to hyperbaric medicine? Nanotechnology is the creation and utilization of materials, devices, and systems through the control of matter on nanometer scale. Nanometer is 1/1000th of meter. That's at the level of atom, molecules, and supramolecular structures. And the official definition by the National Nanotechnology Initiative, USA, is the understanding and control of matter of dimensions roughly between 1 and 100 nanometers. We have to follow this arbitrary definition as dealing with many materials that would undergo some regulatory approval process, and they are classified as nanomaterials if they fall between 1 and 100. And nanobiotechnology is simply the application of nanotechnology into biotechnology. It's a mixture of these two, this term. And another term, nanomedicine, is the application of nanobiotechnology to medicine specifically, and not just all the other biological sciences. And even further, you are going to hear terms such as nanoneurology, because this prefix nano is just going to be used for many other branches of medicine. For example, for cancer, it's kind of the term nano-oncology that has seemed to catch on. Then diagnostics. Molecular diagnostics has been around for almost 20 years, but it has been refined now with the introduction of nanobiotechnology. We can call it nanomolecular diagnostics, because all the nanoparticles can be used to detect single molecules. They can be used to detect single microorganisms, single viruses, bacteria. Bacteria are much bigger, but viruses being only a few nanometers, they are a problem in detection. And here you don't have to amplify the PCR, the classical technique for molecular diagnostic polymerase chain reaction will depend upon amplification. But here you can directly detect pathological organisms. So nanodiagnostics for neurological disorders is a very important part of the therapy of neurological disorders, particularly the personalized therapy, which combines diagnostic with therapy. And incidentally, of course, biomarker detection is refined, and many other techniques, such as PCR that I mentioned, has also been combined with the nanoparticles. So it has been refined as well. So you may go through the steps, PCR, but your detection limits are much more gone to a lower scale. You can detect things you couldn't detect before. And there are all kind of biochips that are now on a nanoscale, use nanochips. And same way, there are nanosensors available that can detect substances and nanoparticles or traces, very minute traces of substances. And I think with all these activities, it will have an impact on the diagnostic relevant to hyperbaric medicine, because we still do a lot of measurements and we still have to use diagnostics combined with treatment with hyperbaric. So this is just for your information that there are a lot of new development. This is an area of great interest to me, and a lot of things are being available. Now we talk of neuroprotection. We have mentioned hyperbaric oxygen in connection with neuroprotection, both for injuries to the nervous system, strokes, whatnot. And nanoparticles are themselves, some of them, as neuroprotective agents. There's a lot of misconception about nanotechnology, nanoparticles being toxic, hazard in the environment. But all the nanoparticles, there are hundreds of different kinds depending upon the substance in which they're made. They're not all harmful. Some, strange enough, are neuroprotective. And the neuroprotective function can be combined with other substances. For example, we can have a drug that has a nanoparticle that's going to help to attach it to the receptors and certain type of lesion so that we can diagnose it, and the drug attached to that can also be used for treatment. And so we are able to have controlled delivery of substances across the blood-brain barrier. And another reason I mention nanotechnology is in combination with hyperbaric for regeneration. Now, in addition to neuroprotection, I mentioned regeneration of the nervous system is very important. These two can be combined, so you don't have to isolate, say, well, this we are going to only use neuroprotection, or we are only going to do neuroregeneration. In a multidisciplinary approach, we may be initiating all these things together. Now, it's a very big topic of neuroregeneration, of all the materials that are being used to regenerate. And of course, nanofibers are used as the substrate, which you can add on. They're like struts. You can add on stem cells to try to repair a lost part of the brain. Let's say a big hole left by lost brain tissue from traumatic brain injury, and you are going to repair it with nanofiber struts. You add on cells. You can facilitate the whole process also with hyperbaric oxygen, because this is going to facilitate not only the stem cells but also the regeneration. So I think these are the basics on which one can induce. So I mentioned already the nanoparticles that are neuroprotectives. Just to identify them, they are Sierra nanoparticle, yttrium oxide, fullerenes, that they can not only protecting the brain from various insults, but some of them have free radical scavenging properties. Now, we talk about free radical damage and talking about how we can sometimes use free radical scavengers in addition to hyperbaric oxygen if you are concerned with enhancement of free radical release, or we are treating conditions which naturally are accompanied by release of free radicals. And our multiple approach to them, beside hyperbaric oxygen, we also want to give something for the free radicals. So this is why it's good to know that fullerenes, carboxyfullerenes, they have antioxidant and anti-excitotoxic properties. And so I think they can be considered this kind of, for this action as potential use against, or if you think that very excessive and high doses of HBO that may be required for certain indications, particularly we're talking about infections and whatnot. If you want to protect the normal tissue against oxidative stress, you might consider. So that's the reason why I mentioned that. The future prospects. So we think nanobiotechnology will not only be neuroprotective, but play an important role in neuroregeneration. And within the next, we may see within the next decade, we have also ability to deliver. Let's say we want to deliver neuroprotective agents directly to the brain, but we have the blood-brain barrier. We can get across that much easier with nanoparticles. And as I mentioned, in particularly head injuries, with the loss of brain substance that may have important. And actually in brain tumors already some systems are in development. They are combining diagnostic with therapeutics, and diagnostics using nanoparticles. And there are some nanoparticles that can duplicate as therapeutic agent as well. So it's combining diagnosis with therapy. This is just a table from one of my, I think it's a book on regenerative therapy. I have the chapter on regenerative medicine, published this year. These are all the strategies that use to regenerate the brain. You can see in red, they're all in some these actions related to nanoparticle. And this one, use of stem cells, along with that, can be enhanced by HBO. So somewhere there is a connection. But this is to give you a general idea of what's going on in this field, and if you're going to be treating such patient with adjunctive hyperbaric oxygen. Now we come specifically to the connection with cell therapy. At the moment, cell therapy is being used for the prevention and treatment of human diseases by administration of cells that have been selected, multiplied, pharmacologically treated outside the body mostly, but the scope of cell therapy is broadened to include methods, pharmacological as well as non-pharmacological, to modify the function of intrinsic cells of the body in vivo for therapeutic purposes. So it is not just only that we are going to bring in the cells from outside, take them out, process and reintroduce them. We can also enhance the multiplication and movement of in vivo cell. What's the aim of cell therapy? We're trying to replace, repair, or enhance the function of damaged tissues. And most of the cells, there are many types of cells used for this purpose, derived almost from any part of the body. Yeah. So apart from the action of, apart from using cells from various parts of the body, you can use cells derived from muscle tissue, you can try to repair the muscle injuries, you can use cells from the myocardium, and so on and so forth. There are many cell therapy based on that. But the most activity of focus is on stem cells. You probably know from what you hear is mostly stem cells. Intrinsic cells are not differentiated, but they have the capacity to differentiate and go and repair specialized organs such as brain or heart. So most of the stem cells you usually think are in the bone marrow, and only one of the 100,000 cells in the bone marrow is a stem cell. Therefore, we need methods to sort of multiply or to expand the cells. So you take them out from the bone marrow, to separate them, or multiply them outside, and then you can use them for transplantation. But to do this, we have many methods. We can use pharmaceuticals that can expand stem cells. Alternative is to do this process in vivo. We have some drugs which might also expand and sort of mobilize the stem cells intrinsically. And there are the whole list of drugs. I won't bother with that, they're under investigation. But the least toxic of all those drugs, hyperbaric oxygen. A typical course of hyperbaric oxygen treatment increases the number of stem cells circulating in the patient's body about 8-fold. This, in general, the whole. Mobilization is, as mentioned, is much more effective than by pharmaceutical methods, and by causing an oxidative stress. HBO activated a physiological redox active autocrine loop in the circulating stem cells, it's been shown in the mouse model, which stimulates vasculogenesis. And then thioredoxin system activation lead to elevation of epoxy-inducible factor 1 and 2, followed by synthesis of IF-dependent growth factors. So this is the mechanism, one of the mechanisms at least, that has been identified experimentally. I have given as example 5 studies where hyperbaric oxygen stem cells have been combined in animal experiments. Let's say that you take neural stem cells. When I mentioned there are undifferentiated stem cells in the bone marrow, but you have specialized stem cells in various organs as well. You can identify neural stem cells in the brain, and they are in experimental animals, particularly most of the studies are done in rats. They can be isolated, they can be expanded at 2 atmosphere absolute for 60 minutes, and they can be differentiated into neurons. So stem cells are, even though they are neural stem cells, are destined to differentiate into neurons, but you still have to make this process before you can use them to repair the brain. And so using these endogenous cells, they can, or at least they have been shown that you can differentiate them, then you can use them for any other purpose. Then if you take the rats with hypoxic ischemic brain damage and expose them to 2 atmospheres daily for about a week, it's an interesting fact that neural stem cells, they migrate to the cortex and differentiate into mature neurocytes. So as nature has intended the neural stem cells to actually go and repair the site of damage, but that doesn't happen by itself. They just don't move, particularly in the brain, they don't move and they are not enough. But under the action of hyperbaric oxygenation, they have been shown to migrate, and that is in vivo. That was ex vivo study. And another study that you have actually, you can see that astrogenesis by beta-catenin induced down-regulated BMP4 gene, and so the mechanism of neurogenesis has been identified in this particular model. Now, there are stem cells in various body fluids as well. One of the sources, the amniotic fluid. You can isolate another type of stem cell, mesenchymal stem cells, and they are more for repairing some of the tissue injuries, other tissue injuries going out of the central nervous system. Rat sciatic nerve injury model. You can apply mesenchymal stem cells combined with hyperbaric oxygen, and it enhances the peripheral nerve regeneration by suppressing apoptosis in implanted cells, and also attenuates the inflammatory response, because you can get sometime an inflammatory response when you implant these cells. And this is going outside of the nervous system, but relevant to neurology and neurosurgery, but neurosurgery particularly because we have in neurosurgery procedures done on the spine to fuse the bone. Sometimes it is done in connection with removal of the degenerated disc, let's say in the lumbar spine, and in some situations we fuse following that. And the fusion in one study was done with mesenchymal stem cells with and without hyperbaric could normally just the bone grafts are placed. But it's also been shown that if you just use bone graft, you can also stimulate the take up the ossification. The bone has to solidify to be useful. But this is done to try to fuse using mesenchymal stem cells. So HBOT alone, although it improves the bony fusion, but it does not enhance the osteogenic ability of the mesenchymal stem cells unless there is added hyperbaric oxygen. So all these studies are, I think, important evidence for the role that HBO would likely to play later on in human applications. Now, I talked about gene expression. Now, gene expression, the process which information from a gene is used in the synthesis of a functional gene product, usually a protein. That's the basic process. And most of the neurological disorders have a specific gene expression chain, and more are being identified, so we can, by measuring gene expression in a particular disorder, have an idea of the status of the disease that time, then how it is modified by treatments. Most of these gene expression analysis is done by DNA microarrays, biochips, and human endothelial cells in vitro following HBO exposure comparable to clinical treatment, they upregulate the expression of antioxidant, cytoprotective, and immediate early genes. So many of these actions, they can be identified by the expression of certain genes. So why do the antioxidant gene expression gets upregulated? Because it is the body's response to HBO exposure. And there are, if you take stroke, there are over 5,000 differentially expressed genes. It's not just, I would say, single gene disease. There are so many processes taking place during stroke. And if you want to study the effect of HBO on stroke at gene expression level, well, you will have to study the gene expression of so many different genes and correlate them with beneficial effect. So there is a tremendous task. Very little has yet been done. So if one is looking for research projects, there is much to do in this area. And there is another change during a vascular insert. That is the, we already mentioned this enzyme, matrix metalloproteinase, MMP9, which shows up in many kind of injuries and other processes. But some of the sequelae of the cerebral ischemia are associated with the overexpression of this. Incidentally, the same sort of thing is also coming in non-healing wounds. I already mentioned with TBI and unstable plates in atherosclerosis. We talked about the danger of having a plaque in a carotid, and we sometimes want to know how unstable it is. Perhaps there are some gene expression patterns where we can look for the expression of this. So there has been a study done that after trauma of surgery, you can measure MMP9 in urine and it's raised, but reduced if you expose to HBOT at 2.5 ATA for 5 times 1 hour a day. So this could be just like a biomarker, which is going to show you some kind of a measure of the effect of HBO. So we are going to have MMN9 as a indicator. Now, there are many chronic vascular pathologies, again, where overexpression of MMP9 gene may result in cerebral vascular accidents, but it can be improved by a series of HBO treatments, which also reduce MMP9 expression. So you can correlate the improvement with the gene expression pattern. So it's another dimension to try to evaluate the effect of. So an understanding of how HBO therapy modulates MMP9 gene expression may provide an additional insight into mechanism and future potential therapies. So I was just mentioning again the application of neurosurgery. HBO, we talked about, I think there was a case of trying to treat cervical spondylosis, but there the focus was on the neurological part, where the cervical spinal cord and anterior nerve roots, because in spondylosis the problem is bony as well as neural structures. Neural structures producing pain and neurological deficits, and the bony things producing more spinal instability or pain, also pain of bony origin. So we have to sometimes fix both. So a nucleus pulposus from the degenerative disc, they produce inflammatory factors. That is known already. Many cytokines such as interleukin-1, prostaglandin E2, and nitrous oxide and MMP are also have been identified in the degenerative spinal discs. And HBO treatment of the nucleus pulposus cells shows decreased expression of interleukin-1 beta and increased gene expression of type 2 collagen, suppression of the phosphorylation of p38 MAP kinase, decreased nitric oxide, PGE2 and MMP3, as well as a raised expression of a tissue inhibitor of matrix metal. Now that is a marker of damage, but there is a tissue inhibitor. The raised expression will be beneficial. So this has been shown that HBO treatment can produce these changes in a disc. So this sort of leads to determining the ratio of anabolic factor, the positive things, catabolic factor, negative things. Ratio may provide a therapeutic approach to slow the course of intervertebral disc degeneration. These are just experimental bases in biotechnology, but you can think of where they may possibly have clinical connection in treatment of degenerative disc disease, potentially. This has clinically not been tried. Gene expression, now from diving, you're mostly interested maybe in decompression physiology. So use of HBO to expedite decompression from saturation is not a proven thing. You don't necessarily have to use high pressure oxygen in the procedures, and there has been a concern whether this will increase the risk of toxicity to the pulmonary system. And in a study on swine and pigs, compressed to 5 ATA for 22 hours was to achieve saturation, and then you decompress them in air or HBO starting at 2.38 ATA. and you can all sacrifice and take out the RNA, ribonucleic acid, from the lung samples, and then go with Affymetrix microarrays. Gene expression patterns were similar in both the air and hyperbaric decompression groups. Neither group developed type 1 DCS, but although some animals in HBO group had gait disturbance. This would say that I don't think, don't believe HBO during decompression reduces the incidence of decompression syndrome. But here is a little disturbing thing we need to be investigated. There was a slight problem with the gait. Could it be that it was doing some damage? We talk about biomarkers. Biomarker is simply a characteristic that is objectively measurable and evaluated as an indicator of a normal biological or pathological process, a response to a therapeutic intervention. I have a book on biomarkers which lists all the biomarkers in different systems under different conditions or diseases. Now, biomarker is nothing new. I mean, in a way, if you check the blood pressure of somebody, that's a biomarker, but they are gross biomarkers. In this age of molecular biology, we're talking of molecular biomarkers, much more refined, and this is why many of these biomarkers are either proteins, DNA, RNA, and sometimes it's the expression of a distinct gene that can be used as a biomarker. And it can be used for identification in a tissue where it will be expressed specifically but not in the surrounding tissue. So this will mark it out as a specific biomarker. So several biomarkers of various diseases have been discovered. So this book could fill with thousands of biomarkers, but very few of them have been validated. A biomarker has to be validated, proven, and that sometimes require trials to prove its value and reliability. Of course, we always see biomarkers in various diseases, and the same biomarkers may sometimes be found in another disease as well. So we have to show the specificity for that condition, and we have to show how it changes. And once a biomarker is validated, it has value as a guide to clinical trial of drugs. There is a tendency in clinical trials now to monitor the efficacy of drugs that are under investigation by measuring biomarkers. Biomarkers are much easier to measure more frequently. It's not a little more complicated procedure like trying to image them in clinical trial, which are going for a long period, require frequent measurements, and it's not practical to have more complicated methods. Biomarkers are very convenient. If they are blood biomarker, just need a blood sample. But here is the idea now. We, for monitoring HBO therapy, this is an area that we should be looking into, to have biomarkers to monitor the progress and the efficacy for HBO therapy, same way as we do it for other drugs. So here is biomarkers of hypoxic brain damage. At least we have something to start on. and it combines clinical biochemical imaging and electrophysiological investigations to approach the outcome. In this particular approach, we could be using biomarker as well, because patients, let's say somebody in a cardiac arrest was remains afterwards in a vegetative state, one of those global ischemia, hypoxia thing we talked about, and if they have a higher level of serum neuron-specific enolase, it's one of the biomarkers, that is usually associated with those who are remaining in vegetative state for a long time. So that's kind of one connection. These are biomarkers of protein destruction. And another biomarker is neurological disorder, S100B. They are elevated in many conditions characterized by damage, and particularly in, of course, in hypoxia. But we will have a lot more work to do to get the specificity for these. Now, we talk about assessing the effect of HB on multiple sclerosis. Now, MRI is the standard measure for evaluating effect not only of HBO, but many of the drugs used for that have been developed for multiple sclerosis use MRI, even clinical trials. But it does not evaluate the level of remyelination within the brain. You can look at, for example, the size of a lesion, but going on a much more refined level, that we need to develop better biomarker. A fluorescent molecule, 1,4-bisp amino sterol 2-methoxybenzene, or let's say for short call it BMB, binds selectively to myelin. And levels of BMB binding differentiates remyelination in shadow plaques from either demyelinated lesions or normal appearing white matter on PET scan. So here we go one step further to determine myelination status beyond PET scan. So future approaches to multiple sclerosis should integrate clinical and imaging data with pharmacogenomic and pharmacogenetic databases to develop prognostic profiles of patients, which can be used to select therapy based on genetic biomarker detected on blood examination. So these two terms, pharmacogenomics and pharmacogenetics, are relevant to personalized medicine. They simply mean that we collect a lot of information about a relation between certain gene patterns and response to certain therapies for certain diseases, and are there certain type of genes associated with ill effects, certain type of gene that can predict a better efficacy, and some or other. This information can be sort of kept stored in databases, and we can pull it out to try to design specific treatments, so-called personalized treatment for multiple sclerosis patient. And let's say take one type of multiple sclerosis, relapsing remitting. It has a specific fingerprint, so to say, of cell-free circulating nucleic acid as specific DNA sequences that provide significant clinical sensitivity and specificity for monitoring response to treatment. So a biomarker like this can be tested in the blood, and you don't have to go and do the MRI scan or any further refinement of it. So there you can monitor the response to a treatment. there are several biomarkers in the CSF. Sometimes one biomarker does not give you enough information. You have to use a panel of biomarkers, and it's like a pattern, a fingerprint. And you have CSF biomarkers such as interleukin-6, nitric oxide synthase, osteopontin, vitamin A. They can be used for detecting responder to treatment in stratification clinical trial, because those who respond to treatment will more likely have these biomarkers. And if you're going to stratify a selective clinical trial, not just keep on giving some same treatment to all the patients, and if you select those with these biomarkers, your results are going to be much better. That's what I mean by sort of a personalized medicine trials that are leading to personalized medicine. Neuroprotection and biotechnology, as I mentioned already, some of these things that we can combine neuro regeneration, protection against neurotoxins, and intervention to slow the progression of disease, all in one strategy. And it doesn't have to be specific, only protecting the brain. It goes beyond that. The classical neuroprotective agents have been hypothermia, HBO, and barbiturate-induced coma. They are still here and there, can still be used. HBO much more than the other two, and they are sort of many other biotechnology-based drugs that I mentioned earlier that include neurotrophic factors, for example. They are being investigated as neuroprotective agents. So the molecular mechanism of neuroprotection by hyperbaric oxygen cerebral ischemia. Now we have already assumed and seen the protective effect in experimental models and with imaging studies, but at a molecular level, that the HBO therapy leads to activation of ion channels, inhibition of HIF1 alpha, and a number of other things. I think I already mentioned these. These were actually part of the biotechnology part of my presentation. So, but these factors that I mentioned before really are detectable by the biotechnology method. So if you're going to investigate the molecular basis of HBO therapy, and you will have to get into method to determine all of these things. And overall, this will help to strengthen the clinical evidence that now in the last minute or so, I will quickly tell you what personalized medicine is: the prescription of specific therapy best suited for an individual. Say, right drug for the right patient. Is usually based on pharmacogenetic, pharmacogenomic, pharmacoproteomic, and pharmacometabolomic. There are other two omics. There are actually many, many more omics. That's just simply proteomic and here metabolomic used for this purpose. I believe the scope of molecular medicine is much broader than just some people call it genomic medicine. I don't agree. It's not so restricted to genes. We take environmental factors into consideration, and they may influence the response of a patient to a certain treatment, whether it's drugs or hyperbaric oxygen. So you have to sort of look at it in a very more broader way, and that's why we call it, another term being used, systems medicine. that allows you to integrate new technologies and translate them into clinical application for improving health care. So, what's personalized hyperbaric medicine? Actually, we talk about personalized medicine more in recent years, but personalized medicine was being applied to hyperbaric medicine much earlier, in the early 1980s. The very fact that the response of stroke patient to HBO was assessed by using SPECT before deciding on further treatments or whether to treat them or not. This sort of decision is called personalized medicine. And it's very similar now to the use of diagnostic biomarker. They are more convenient than SPECT, and we just want to have some proof of efficacy and not just keep on treating a patient even when there is no improvement, or we discover that after wasting many treatments. So the difference now is in the post-genomic age, we're looking for genes as biomarkers of response to HBO as well as predictive of oxygen toxicity. So the next step would be, would it be possible to predict who is more liable to develop oxygen toxicity? It is not simply a matter of dose. You can say, well, above this ATA, you're going to develop oxygen toxicity. The individual has other factors that are going to determine, and they may be genetic factors, and you can perhaps be able to predict oxygen toxicity. This is a thing that